Move to our next speaker. So next up is Chloé Delepine, that she will talk about the contribution of astrocytes to motor learning. And Chloé is a postdoc in the lab of Mriganka Sur at MIT. Stage is yours, Chloé.
Thank you. Yeah, it's been an amazing session so far, and I'm gonna finish it with yet another perspective on the motor cortex, and this time focusing on the astrocytes' contribution to motor learning. Oops. Okay. And so astrocytes, as you may probably know already, are cells that are tiling the brain and that contributes to brain function via multiple ways. That has been evidence for their role in metabolism, blood-brain barrier, synaptogenesis, immune response. But what I'm particularly interested in is to study their role as responding and regulating neurotransmission, notably through neurotransmitter clearance. And there is evidence also that astrocyte responds to neuromodulators via expression of G protein coupled receptors and calcium signal. And all of this, there is evidence for all of these functions, but however, there is only a few in vivo studies that have been focusing on the astrocyte neuron interaction during behavioral task and learning. And so that's what we decided to focus on for this postdoctoral project. And I've been decided to choose motor learning as one of those complex behavior to focus on. And so motor learning, in my definition of it for this project, is the acquisition of a reliable and dexterous movement. And it's been shown that the M1 cortex, as you've seen before, is involved in motor learning, including the acquisition of the task and also the performance of the movement. Motor learning is associated, at least in rodent, with the emergence of a new ensemble of neurons, which are a subset of highly correlated neurons that are active during the learn movement. And we decided to use this quite simple lever push task that was described by the Komiyama lab first, where a mouse is receiving a reward when it pushes a lever to a certain threshold after a cue tone. And we've been asking the following questions. So first, we know that neurons are functionally remodeled during motor learning. What about astrocytes? Are they also changing during motor learning? Can astrocyte modulate motor learning? And can astrocyte modulate the neuronal changes associated with motor learning? And to answer those questions, we apply various strategies. Okay, so first, are astrocytes changing during motor learning? To answer that question, I've purified astrocytes from M1 cortices of mice that have been trained to different stages of the learning, so either naive, novice, or expert mice. And I've discovered that around 100 gene sets are indeed changing their expression during motor learning. And here is the most dysregulated gene sets that you can see. And what you can notice is that most of them are sets of genes that are transporters, transmembrane transporters, and most of them are overlapping by including glutamate transporter genes, such as GLT1 and GLAST. So that's the first thing. Then Jennifer, with also a postdoc in the SIR lab, has been looking at calcium signaling in astrocyte microdomain using in vivo chronic imaging of mice trained to this same lever push task. And our main result is to show that in an expert mouse compared to a novice mouse, you see an increased coincidence of calcium events in the microdomain during the push, during the lever push. So we've been showing that yes, astrocytes are changing during learning. They show some kind of plasticity that include probably other things, but we identify that it's including glutamate transporter gene expression changes and astrocyte calcium activity changes. So then are astrocytes able to modulate directly motor learning? To answer that part, we decided to target astrocytes using genetic and chemogenetic manipulation of their functions. And we use two different models. The first one is to knock out the glutamate transporter GLT1. So it's the main glutamate transporter expressed in the cortex and mostly expressed by astrocytes. The other tool we used is to use an engineered GQ-GPCR receptor that can be activated by an exogenous ligand, CNO. And so this was used by, using flux GLT1 mice with local injection of the Cre directly in the motor cortex. And here we directly inject the viral expression of the GQ dread. So what are the effects of this manipulation on behavior? First, for the GLT1 knockout mice, we showed that actually the mice are still able to learn the task normally. So this is their hit rate, this is their response time. It's similar to wild type. However, if you look at the lever push trajectory that are shown here, you can see that the GLT1 mice do not learn a dexterous movement. Their movement is not stereotyped nor reliable. And the GQ dread animal, the GQ activated animals, on the contrary, and so if it was not clear, this GQ is specifically expressed in astrocytes, of course. So in those animals, we see actually that both the performance and the movement dexterity are impaired. So slightly different result than the previous model. And in this model, we were able to try to inject this controlled saline solution instead of the activating CNO. And we see that in that case, actually, you can rescue the performance. So yes, astrocytes seems to be able to modulate motor learning, and we showed that knockdown of GLT1, so glutamate clearance, decrease of glutamate clearance will trigger an impaired dexterity, but will not impact the timing, the response time of the animal. However, GQ activation of astrocytes impair both the dexterity and the timing. So then we focused on neuronal activity and with the same two models, assess what were the effects on neuronal activity. For that, we used in vivo chronic 2-photon imaging again, but this one of M1 neurons, and by using GKM6S calcium sensors. So with this technique, you can image hundreds of neurons simultaneously during the performance of the task. And so we looked at that in expert animals. And so from what we can see with looking at calcium sensor fluorescence, is not much difference in the activity itself, but what we could see is a decrease of the interneuronal signal correlation during the movement period. So that's what you can see here. More neurons are with low interneuronal correlation compared to in wild type, where you have at least a group of neurons that are highly correlated. In the GQ activated mice, again, not much change for the activity, but we see, on the contrary to the GLT1 mice, we see an increase of the interneuronal correlation. As you can see here, there is a shift for more neurons being highly correlated. So then we wonder if we could look at how the motor cortex neurons are encoding different task parameters during this task, and if we could see differences in the encoding of the task parameter looking at our two models of astrocyte modulation. And this was work done in collaboration with KG Lee, who's been running all those modeling. And one thing that we've been looking at is using GLM models and measure their performance to predict single neuron activity from those different task parameter. So that include trial event, like the start of the trial, the reward time, also the lever movement, so the trajectory, the speed, and finally, if the trial was a success and the response time. So when you take all those predictors together and you look at the performance of the model, you observe that both the GLT1 and the GQ animal have a lower performance of the GLM to predict the neuronal activity. But if you look at the contribution of each of those variables, first to the performance of the model, you can see first that in wild type we identified that the parameters that are the most encoded or the most contributing to the neuronal activity encoding are the success of the trial, the response time, and the speed of the movement. So that was the first interesting result. And we see actually that in the GQ animals, it's mostly the same contribution. The distribution of the contribution of the variable is mostly the same. But if you look at the GLT1 animals, it seems that the response time is contributing proportionally more than the other variables. So that was interesting. And actually, so if you remember the behavioral result that I show you, this could be a nice representation or nicely associated at least with the fact that the timing in those mice, the response time was not impaired. Only the dexterity of the movement was impaired though. So that's interesting. So yes, we can see that when you modulate astrocyte functions and notably the glutamate clearance and the GQ signaling, you do see a change in neuronal correlations. And you see a decrease of the neuronal encoding of task parameters, notably the response time and the movement speed. So in conclusion, M1 astrocytes do show in vivo plasticity during learning of a lever push task. And we've shown that by transcriptomic and functional analysis, astrocyte manipulation of M1 are sufficient to alter motor learning and execution of the movement, as well as the neuronal population coding, again in the lever push task. And finally, we demonstrate that astrocytes have a specific role in coordinating M1 neuronal activity during motor learning and to control the learned movement execution and dexterity through a mechanism that seems to include fine regulation of glutamate transport. And with this, I want to thank you for your attention and all the members of the SIR Lab, my collaborators, and financial support. And I'm happy
Thank you very much, Chloe, for also being exactly to the time, perfect timing, and great talk. So while I'm again waiting for the questions, I'm gonna actually ask you to a little bit elaborate on the interpretation of so the manipulations you performed that you showed us that are partly different results depending on the in the GLT1 mice or the GQ mice, I think, that when you use DREAD or and then also the correlations that are like one is decreasing the correlations in the neural activity, one is increasing. Like how do you put this together and then the impact that they have on behavior, on the motor behavior?
Yeah, the parallel between the two is very interesting, and we are still trying to figure that out. But so yeah, it seems that the two models are opposite in some ways. So GLT1 being decreasing the correlation, GQ DREAD activation seems to be increasing the neuronal correlation. But in other way are the same. Like in both animals, the animals are not learning well to do a reliable movement. So there are some overlap and some opposition. So we are trying to look at that by, so for example, we started observing that the GQ DREAD animal also show actually an increase of the expression of the GLT1 gene. So there could be some molecular mechanisms that are indeed opposite in the two models. But also my global idea of this is that if you perturb the motor cortex you will, so the correlation of neuron in the motor cortex is the very important part of the motor cortex function. And so if you dysregulate it, if it's too correlated or too less correlated, in both cases, you will prevent motor learning in some ways. And then it seems that the response time is affected differently. And for this, we don't have any mechanism to suggest yet, but that's very interesting that in one case, the response time is preserved and not in the other case.
Thank you very much, Chloe. Well, I have one more question. I think we have quickly, if you could shortly answer this question. So the question is from Gregory Handy, and great work, Chloe. And what are your future steps in investigating these pathways? For example, can you do GLT1 knockout and GQ dread at the same time?
Mm-hmm. Yeah, it's a question that I've been asked several times now, and it's definitely in my plan. Actually, I've started gathering some mice to do this. It's gonna be my next experiment. Yeah, thanks.
Okay, thanks for the great question and great answer. And with that, I would like to again thank all the speakers.